This is a short video just on Chapter 4 and the sections within that that are most important for our next unit. We are going to be starting to talk about macroevolution, and this is really looking at patterns of speciation over over many species. So looking for trends across species, across families, across orders. We are going to start to apply phylogenetic thinking now back in evolutionary time, and we're going to be looking at evolution using evidence from the fossil record. So what are our expectations for patterns we would see in fossils based on what we know from phylogenetics? For instance, if we know that certain taxa originated a long time ago, we would expect that we would find fossils from those taxa in older fossil deposits than species that we think originated more recently. I'm going to call your attention to figure 4.16 here in the text, and I want you to ignore for the time being part A because it shows you that not the expectation. It shows you something that doesn't conform. So here on the x-axis we have the time of appearance in the fossil record. So as you move to the right, you find these fossils in older deposits. On the left-hand side, this is phylogenetic branching order. So using this little phylogeny here, D gets number 1 for what they call clade rank, which is it is on the first branch in this particular phylogeny. So for instance, when you look for fossils of D compared to fossils of A, you would expect the fossils of D, because it has this earlier branching time, right, 1 versus 3, that it would be found in older deposits, right? So we would expect to find D somewhere over here on the right-hand side and A somewhere over here on the left. And if these things were perfectly correlated, you would expect to see this straight line here, like you do with on B. These are 13 clades of horses, equids, and then on the right, dinosaurs, and 12 clades, and it more or less shows the pattern. You can tell how closely this pattern conforms to our expectation by how close this letter S is to 1. The closer it is to 1, the more correlated, and so obviously the horses here are highly correlated, where the dinosaurs are not quite as correlated. Okay, let's take a look at, sometimes this isn't true, right? Sometimes you do get this pattern, this confusing pattern they tried to show you in A, where you don't get this match, or for instance, the dinosaurs weren't a complete match. So when would this expectation not be met? What are the conditions under which that might happen? Well, for one thing, we know that the fossil record is incomplete. So many times the fossil record will be incomplete, and we won't be able to fill in all of those dots that would give us that pattern. Also, there can be times in evolutionary history where when you look back at that clade and you look at D, that species that is on that early diverging lineage, it's possible that the characters that you would use to identify that species, those characters originated way after that branching actually happened. And so the modern D doesn't look like what its ancestors looked like at that early branching time. And that does happen. There are many examples of that. So we can have these expectations for fossils, but we also know that those expectations can be broken. I asked you guys to take a look at Cope's rule. Here is Cope's rule. So if we start looking across taxa in the fossil record, right, we're at macroevolution. So we want to see in particular locations or in particular time periods of evolutionary history, do all the species or many of the species all kind of do the same thing, even if they are distantly related, not closely related to each other. And these are parallel trends that we are looking for. Cope's rule here is that in many animal clades, body size has increased over time. So here you have the present, here you're going back in time, and you can see that early horses were small. And they become bigger and bigger and bigger. There are a few exceptions to this rule, but for the most part, especially in horses, horse taxa got bigger over time. Okay, so now we want to start thinking about how do new species form? Well, we know that we identify new species based on their morphology in fossils, and that if they are physically different, we might call them different species or different taxa. So when we ask how new species form, we're actually kind of asking the question, how does morphological change occur? Um, is it rapid? Is it slow? Is it gradual? And then the same thing for speciation. Is that rapid or is that slow? And then are those two things correlated? Do they occur at the same time? Does morphological change and speciation happen at the same time or not? And there are a number of hypotheses. We're going to focus on a couple in this class. So here, let me orient you to these graphs, and they're all basically the same, where you have some measurement, like, we just did size in horse. So for instance, size is increasing going to the right here, and time is increasing this way. Okay, so for instance, your body size is getting bigger as time passes. Right, here's more recent time. So these are just some measurements for size. So let's take a look at B here, and you can see that increase is happening over time, but that pattern is kind of gradual, right? And it doesn't tell you on here where a new species forms. It just basically says no new species is forming. There's change, but it's all within the same species. And that we call phyletic gradualism. So gradualism makes sense there. We have another occurrence in which change happens but is not associated with speciation, and that's D. So we're going to go from B now down to D. You can see no change, no change, no change. Lots of change, lots of change, lots of change. No change, no change, no change. So this is stasis, right? And then lots of change. And this is called punctuated gradualism. So we have phyletic gradualism, where it's sort of gradual, and then we have punctuated gradualism, and that's where the change is punctuated, right? Rapid, then slow, rapid, then slow. Okay, so now in neither case here did we have speciation. Over here we have punctuated equilibria, which is very much like punctuated gradualism, right? In the sense that you have stasis, and then you have rapid change, stasis, rapid change, stasis. But what they're pointing out here with this forking diagram is that here speciation is actually occurring. So you have this stasis going on here, this lineage, no morphological change. But over here, lots of change, and this is a new species. And so in this case, rapid change and speciation are correlated. They are happening at the same time. So no speciation here in this red. This is just as if you were measuring some trait, right? This is the peak of that trait, the mean value, and it's the same. But in this taxa where they're representing the value by black peaks, you can see that the height of that peak, that mean, is changing over time. It's shifting to the right. And that's sort of rapid. Depends on what this scale is for time. Okay, so over time this is changing, and this is a new species. Now we have stasis again. That trait value is not changing. And then in this, there's a speciation event right here. One lineage not changing, the other, this new species here, is changing. So we have two new species, one with the trait value of the ancestor and one with a very different trait value. Okay, that's punctuated equilibrium. And then we have phyletic and punctuated gradualism, neither of which are associated with speciation. Okay, so I want to give you an example, actual trait values. So this is a measurement here um, of the of a bunch of characters that are associated with tooth shape in, I think these are moles actually. So this measures the amount of enamel, that's the pink stuff, the cement, which is the brown stuff here, and then the height, right? So up and down of the tooth over time. And what you can see is that the mean value, right, is shifting over time. Mean value for whatever their number is that represents all those different components of tooth change. You can see that it's shifting over here to the right as time is increasing. We're going back in time from present to 3 million years ago. So in the present day, we have this higher value of the tooth, right? It's taller, it has more cement, more enamel, or yeah, more cement and maybe less enamel, I guess. And then back in time, right, we get shorter teeth, less cement. Okay, so this one, think to yourself for a second, we're not showing speciation, right? So this is going to be gradualism. So let's go back for a second. Is this phyletic or punctuated? And you can kind of see it just as meandering, right? So we're going to go with this one being, ready, phyletic. Okay, I hope you're able to test yourself there and that went well. Okay, so now I want to give you another example. This one is not going to be gradualism because you can see that we have this branching pattern here, right? So this is morphological change on this x-axis again. So the further apart two lines are means the more different they are in morphology. These are some mosses, and you can see that there's no change, and then suddenly we have we have one lineage that sort of stays the same, and then there's this appearance of a whole new lineage, very different in morphology. And then over time again, that happens again, that happens again, that happens again. And I think they're pointing out here that there are more species than are shown on this diagram in this group, because they're up to 10 species and we only have 5 species on our chart here. So this is showing punctuated equilibrium. Now, how exactly does this happen? If you're trying to figure out in real life, we have these species that are distributed across across a landscape. Eldridge and Gould proposed that this happened because the new species were actually evolving in small populations. They're sort of, parapatric, separated off to the side, and then they migrate into that range where we're actually sampling our fossils. So it appears like they appeared fully formed in our sampling area, when actually they probably evolved, mutated, developed in a different area, and we're just finding the evidence of it after that evolution has occurred. So you might be asking yourself, well, what do we actually see in the fossil record? How much do we see this sort of directional Cope's rule getting bigger, for instance, over time? And how much do we see punctuated change? So not a lot of change, and then boom, really big and different in C. Versus random change, which you see in B, and then stasis, where this trait value stays the same over time. And what they're showing right now, these are real examples in the fossil record of each of these occurring. I just talked to you about directional evolution. This is the conical shape of shells. We talked about the teeth, and we talked about horses in body size being directional, all getting bigger, for instance. What we actually see in the fossil record more often than not is random change, up, down, back up again, and a lot of stasis, no change. So we see B and D here, random and stasis, more often than we see A or C. but we do have examples of all 4 of those things occurring. The very last thing from this chapter that I wanted to point out is that we talked about rates of evolutionary change in molecular characters and how that rate varies across different sites, different loci, different positions on the chromosome, mitochondrial versus nuclear DNA. We talked about lots of different parts of the genome and why they might mutate at a faster or slower rate. We want to also point out that the rate of evolutionary change in morphological features varies as well. So this is a nice chart that shows you the amount of time that has passed since that species was extant or alive, and then the rate of change over here, where high rates are at the top on the y-axis and slow rates are on the bottom. And so you'll notice that data points from lab experiments are on this really fast time scale. We're measuring in units of Darwin, and we're measuring these over very short amounts of time, whereas if you're looking at ancient fossil lineages, we could look at 2 samples and they might be 10,000, 100,000 years apart in time. So our sampling intervals are by nature much, much longer. So even if they're in these fossil populations, surely there were very fast evolutionary rates occurring, but we can't really observe those because that sampling period is so spaced out. So while we know that evolutionary rates can be very high, we also know that they're probably not sustained at that very high rate for very long. Otherwise, you could see some really wild changes over the millions and millions of years that species have been evolving. So that's hopefully going to help you a little bit with Chapter 4 and just getting into the idea of what macroevolution is and how we're going to start thinking about evolution in this unit.